Okay, so we're going to change gears a little bit from talking about circuits that are made up between neurons in the brain to talk about a cell biological circuit, which is basically how the cell manages its proteome. And by way of introduction, I'd just like to remind you of all of the information processing that a single cell does. So a pyramidal cell, for example, in the hippocampus receives around 10,000 inputs. And those inputs are processed independently, we believe. And that's because they come from unique presynaptic cells. So if you think about that, and you zoom in on an individual synapse and ask what is it that allows that synapse to respond, of course, from my perspective, I would say that it's the specific complement of protein that's present at that synapse, how it's wired up with all of its neighboring proteins, and you throw in some post-translational modifications, and you could probably make a good prediction about how that synapse would respond. But what's particularly intriguing to us is the fact that you get in the nervous system both stability and modifiability from these elements, the proteins, which actually have a natural half-life. They turn over. And so in order to think about what I'd like to discuss today, I think it's instructive to sort of consider what kind of proteomic challenge an individual cell faces. And I've just put in some ballpark numbers here. So many years of cloning and 2 hybrid experiments have allowed us to identify probably most of the integral proteins that populate synapses. And we could argue that there's probably around 500 of those. And for many of those proteins, we've been able to establish copy numbers. So let's say there's on average 50 copies of each of those proteins. So that would give us 25,000 proteins per synapse in the membrane and in the cytoplasm. And we have 10 to the 4 synapses per cell, so that's 250 million proteins just in the postsynaptic compartment, not considering the huge volume that represents the presynaptic cell. So if we just wanted to make a conservative estimate, we could say maybe there's 500,000 proteins in a single cell. And as I said, those proteins turn over. We don't know the half-lives of all of those proteins, but recent work suggests that an average half-life for a neuronal protein would be around 24 hours. So that would mean every 24 hours, half of the proteome is turning over, 250 million proteins are made in the cell. How do they actually get to the right place? And this problem is even more daunting when you consider the complex morphology of neurons. It's not just a round cell where you could have a point source of a protein that goes out and binds, but rather we have miles of dendrite and axon that are required and are the sites of synapses, and that's where those proteins need to get to. So how do you actually supply protein in this system? Well, many years of really beautiful biochemical work has told us a lot about the mechanisms that regulate protein synthesis, how cells respond to external inputs and regulate their translation. And those experiments all come from what I would call round cells. And so therefore it's no wonder that for many years we focused on the idea that all of the protein that the neuron needed was actually made in the cell body. Even though there were intriguing observations in the 60s and even in the 80s that protein synthesis machinery is actually present and functional in the processes. So this is an electron micrograph from Ozzie Stewart and Chip Levy's work looking in the hippocampus, where they saw in the electron micrograph hallmarks of protein synthesis, that is electron-dense material that appeared to be polyribosomes, so actively translating machinery. And in these structural studies, they speculated that these polyribosomes might actually be responding to synaptic signals. Here you see a presynaptic nerve terminal generating protein that then would go back to influence the synapse. We got interested in this in some of the very early experiments we did when I started my lab, and we were looking at synaptic transmission in the hippocampus and looking at the growth factor BDNF and examining whether BDNF might also have influences on synaptic transmission in the adult nervous system, following on the work of Anne Lohoff and Mu-Ming Pu, who had studied this in culture. And we discovered that if we applied BDNF to slices, that we could get a long-lasting enhancement of synaptic transmission. And like other forms of plasticity, we were curious as to whether this plasticity required the synthesis of new proteins in order for it to last. And we were doing field potential recordings where the recording electrode is a couple hundred microns away from the cell body. And therefore, we were very surprised when we got the result that application of a protein synthesis inhibitor blocked even the early aspect of this potentiation. So the fact that the synapses we were recording from were so far away from the cell body suggested to us that the proteins that were being made here needed to come from a local source. And we went on to show with lesion experiments that the proteins were not coming from the cell bodies that were in the pre- or postsynaptic cells, but rather were coming from a local source. Now, since this time, there have been many experiments, including the experiments of Christine Holt, who was supposed to work before me, speak before me, and many other labs that have shown that local translation is important for different forms of plasticity at synapses. But given the numbers that I just presented to you, we have wondered if we're not selling this process short by just relegating it to a few cases of synaptic plasticity. And given the task of the neuron to supply synapses with protein, we've considered the possibility that maybe a fundamental source for protein may be a local source. In order to understand this, of course, you would want to have an idea of what the potential is for local translation. And this is some experiments we set out to do when my group moved to Frankfurt a few years ago. And as a starting point, we examined the latest experiments that had been published describing the population of mRNAs that were present in the neuropil or in the dendrites of neurons. And so we, for example, took 3 different studies with the largest numbers of mRNAs that were identified, the Allen Brain Atlas and 2 microarray studies. And we were surprised when we looked at the overlap between these studies that there was no single mRNA that was actually on all 3 lists, and the overlap between any 2 lists was also quite small, which suggested to us that there was a lot of room for discovery here, because if we are indeed getting close to the final list of mRNAs, one would expect to see a lot more overlap. So we applied the latest technology for discovering RNAs to the same tissue where we had originally done our electrophysiology. So we microdissected the somata and the neuropil from the hippocampus and subjected this to deep sequencing. And when we did this, we obtained, after we got our nucleotide reads, we mapped it onto the database, and we obtained some 8,000 different genes in this tissue. Now, everyone who's worked in the hippocampus here will realize that this neuropil here has much more than just the dendrites and axons. And so the genes that would arise from these competing compartments were, of course, a concern to us. And luckily for us at the time, Kristen Harris had, together with Mitya Chlavsky, conducted a very nice volumetric analysis of a chunk of the neuropil exactly from the rat hippocampus. And there they described that the volumetric contribution was around 90% dendrites and axons and 8% glia. And in this study, they deliberately avoided tissue sections that contained interneurons, but of course we need to consider interneurons as well as endothelial cells that comprise the vasculature that supplies oxygen to the hippocampus. So we went through a filtering process where we took advantage of transcriptomes that had been published for each of these competing compartments, and took a conservative approach, saying if we find a transcript that's on this list of glial transcripts, we will subtract it from our list, keeping aside that we have a mechanism for rescuing transcripts, so that we could come up with a conservative list of the transcripts that we believe were truly present in this neuropil compartment. And after we did that, we ended up with over 2,000 mRNAs that were present in the neuropil. And if you look, this is just a subset of these gene families that you find here. You can see that there are the receptors for inhibitory neurotransmitters, excitatory neurotransmitters, signaling molecules. It's kind of a synaptic biologist's wish list for what you would want to build and modify a synapse. So these experiments suggested to us that it may be that local translation is much more fundamental than we had previously appreciated. Now, having obtained the data from deep sequencing, we wanted to go ahead and validate the data. And to do this, we used 2 different technologies. One technology that allows us to quantify with single mRNA resolution the mRNA molecules and also validate their presence. And this technology is called nanostring. And in this technology, you use a fluorescent barcode that's specific for your mRNA of interest. And you have another oligonucleotide that has a biotin group on it. So you can design many such barcodes for a whole family of mRNAs that you would like to detect in your sample, and you mix your sample with the barcodes and the biotin tags that are specific for each mRNA. And then when they both bind, the biotin group allows it to stick to a slide, and the whole thing can be imaged. And then in the end, you're left reading off the fluorescent barcodes for the genes that you want to detect, and you can actually count their abundance. And you can get really nice numbers on this because you have spike-in controls where you know the absolute number of mRNA molecules in the sample. So this is what the data look like. So these are individual mRNA molecules with a certain barcode here, and we can actually read off, if you have the code, exactly how many molecules are present in the sample. And so we've done this for about 300 different mRNAs and validated those 300 mRNAs that we originally identified in the deep sequencing. And not only can we tell that they're actually present in the same starting material, but we get some information about how abundant they are. So here you can see each blue dot represents a different mRNA species, and here you can see CAM kinase 2 alpha, which is arguably the gold standard mRNA, one of the earliest ones that was identified, and by far away the most abundant. And you can see that they range in abundance over 3 orders of magnitude, which we think may represent not the fact that these low abundant molecules are present in low copy number across the entire population, but rather may represent markers for a small population of cells, pyramidal cells perhaps. Now this allows us to validate the sample and to quantify it, but we, of course, still haven't visualized these mRNAs in the tissue of interest. And so in order to do that, we used high resolution in situ hybridization using a new technique that allows us to identify single puncta. So here you can see CAM kinase 2 alpha messenger RNA molecules as well as GluA2 messenger RNA molecules, and these are neurons in culture, but we obtain the same kinds of results when we do these experiments in slices. So here are the cell bodies where, of course, you see a cluster of transcripts because these are where the transcripts are made, and hopefully you can appreciate that the CAM kinase molecules go all the way out throughout the dendrites, but the glutamate receptor mRNAs are much less abundant, but still can be observed in the dendrites, particularly concentrated at branch points. We've done this kind of analysis for now over 200 different mRNAs, and we see these kinds of patterns where you can see in all of these are isolated dendrites and the gene names are here. The cell body is off to the left here. So you can see for some that there's a high concentration of particles that are in the proximal dendrite, but for others there's a relatively low concentration, and that doesn't really predict the extent to which you're going to find something out in the distal processes. So this is another piece of evidence that argues against the diffusion model of mRNA molecules being just diffusing out. In fact, we know that they travel on microtubules, because if that were the case, you would always expect that you would be more likely to see mRNA molecules out here when you had a high abundance here. So based on all of these data, we together, using the validation techniques of nanostring and of in situ hybridization, can say that we have 95% confidence that 90% of that original data set, so around 2,300 mRNAs that we identify, are true positives. So this leads us with a lot of potential for translation in the dendrites and lots of experiments, obviously, to do. One of the things that we're interested in is how the mRNAs actually get out there. So where is the information for targeting? Now, we've known for many years that in other systems where mRNAs are localized, for example, in the developing Drosophila embryo, that information for the targeting of the mRNA is contained in these untranslated regions, which flank the region of the mRNA that actually codes for the protein. And people have shown that different versions of an mRNA can be generated, much like alternative splicing for coding exons, that one can generate different versions of, for example, the 3' UTR of an mRNA by choosing different places within that transcript to end the mRNA and to build the poly-A tail, which is the hallmark of an mRNA molecule. So we wanted to discover then the diversity of these mRNA molecules that are present in these different compartments. And the way that we did this is to use a different sequencing technology. So in the original discovery of the mRNAs, we used the 454 technology because it gives very long nucleotide reads that can span the entire coding and UTR space. But in order to discover the different versions of the 3' UTR, we took advantage of the fact that all mRNAs end with a poly-A tail, and we purified the mRNAs and then sequenced in from the poly-A tail. So this is so-called 3' UTR sequencing or 3' N sequencing. And so instead of ending up with reads that are throughout the mRNA, you end up with a stack of reads at the end of a different 3' UTR, which would tell you, if you had 2 stacks of reads like this, that you would have an mRNA with one UTR that was quite long and another UTR that was short. So we did this looking at the UTRs, and we found some very interesting things. One is that if any of you comb the databases and you're familiar even with the sequencing that's done in human populations, you're well aware of the fact that the UTRs have largely been neglected in database annotation. And so you can see here that for most of what you can find in the database, if you look at the number of UTRs that are annotated, for more than 50% of the UTRs have or transcripts have a single UTR. And for our data, which are shown in purple, you can see that we dramatically expand the number of UTRs per gene. So this is just sort of being one of the early people to sequence this. It's nothing special. It's just undermined data, meaning not mined data, not undermined data. The other interesting thing that we see is that there's a big expansion in the length of the UTRs. So if you look at what's in the literature, most of what's annotated at least is very short UTRs, which of course, if you think of the UTR as what you can add to the mRNA, you add regulatability by adding into this thing, into the UTR, we dramatically add the potential for regulation in our UTRs. Now, another interesting thing that we've done is to look at the UTRs across evolution and also across tissue. And you can see one of the striking things, of course, in the genomics era was our expectation that we were going to have a dramatically larger genome than other species. And in fact, we learned that we have the same number of genes as a fruit fly. And many people have argued that it's protein interactions that are important, that diversify us and make us who we are. But another important difference, of course, is the regulatability of translation. And here you can see across from plants to mammals to primates that there's an extension in the average 3' UTR length. And this is just looking in existing databases. Moreover, if you look across tissue, you see that brain tissue typically has longer UTRs. And then if we look at our data set, comparing the transcripts that we see in the cellular somata and the neuropil, we see actually that the neuropil has the longest 3' UTRs when compared to somata. And this is actually a much larger difference than you might imagine, because keep in mind that the somata, when we sequence the somata, we sequence both those transcripts that will stay in the soma as well as the transcripts that will eventually be trafficked to the neuropil. So this bar here is contaminated by the will-be neuropil transcripts. So this is interesting to think about in terms of the diversification of mRNA translation. And if you actually zero in and ask if you compare genes for which you can only find a single UTR to genes for which you see multiple 3' UTRs, and you ask what gene families are represented by those two groups, what you see is that genes that have multiple 3' UTR isoforms are in the categories that presumably really interest people here in the audience, which have to do with synaptic function, neural communication, and the genes that have a single UTR isoform, which means there's really only one level of regulation that's built into that gene, are genes that more fall into the category of what we would consider housekeeping genes. Moreover, if you compare the neuropil to the somata and you ask how many 3' UTR isoforms can a given gene have, what we see is for localized transcripts, these are the transcripts that we originally identified as being present in the neuropil, they're much more likely to have multiple UTRs than the transcripts that are present in the somata. And not only that, the transcripts that are present in the neuropil are much more likely to make use of the distal poly-A site. So that is to be a transcript that has a long UTR, so maximizing the regulatability of the translation. So how is this accomplished? So we have a gene that has within the UTR several different sites that can be chosen for polyadenylation. And these polyadenylation sites are characterized by a hexanucleotide sequence that has a lot of A's in it. And what is typically found if you look at the genes that have multiple sites is that often this last site has the so-called canonical polyadenylation site. It's AAU, AAA. And these guys will have a slightly degraded but still usable poly-A site. So depending on which one of these sites is chosen, again, you can build an mRNA that has a different length of a platform. Now, how is that accomplished? So during the processing of an mRNA, as I said, you could have a non-canonical polyadenylation site, so that would be not the AAU, AAA, and here would be the classical one. Now, normally, both of these sites would have access to the machinery that would ultimately cleave the mRNA and result in the polyadenylation. But if you have, let's say, a transacting protein that binds here and masks this site, then you could favor, for example, this site. And indeed, such proteins exist, and one of the proteins that I'll tell you about now is one called poly-A binding protein, nuclear protein, and it binds to these non-canonical sites that are typically associated with the shorter UTR and favors then the choice, the selection of the long UTR. And in other cells, people have shown that knockdown of this protein leads to a decrease in long UTRs. So when this protein is absent, the non-canonical site is more frequently chosen. This protein is present in hippocampal neurons and is abundant in the nucleus where this event takes place. And what we wanted to do was to then first search and see within our data set how many potential targets are there for this kind of regulation. So when we look through our entire data set and we asked that there were two requirements for a given transcript, it had to have at least two different 3' UTR isoforms. It had to have a long version that had the gold standard poly-A site and a short UTR with a non-canonical poly-A site. And when we did this, we found that there were 1,200 potential mRNAs that fit these criteria. And then when we looked at the categories for these genes, we again see that they're sort of nestled, these are the top 15 categories, nestled in these synaptic genes that are important for synaptic transmission and the regulation of synaptic transmission. And so these particular genes are genes that we've looked at to see if the poly-A binding protein might actually regulate the 3' UTR choice. And I'll tell you about one such set of experiments. So what we've done is to knock down the poly-A binding protein either using short hairpin RNA or actually an endogenous mutant that gives rise to ocular muscular dystrophy. And this mutant actually is a functional null. It sequesters the protein and prevents it from binding. And so we've asked what happens if, I'll show you different experiments using either of these things, if we knock down the poly-A binding protein. And here's results looking with immunohistochemistry against PABPN1, showing that if we transfect cells with a short hairpin RNA, we can successfully reduce the concentration of poly-A binding protein to about 60% of its normal levels. And then what we're going to do is we're going to ask, and I'll go back here, we're going to ask if we get rid of the poly-A binding protein, the prediction is under normal circumstances, poly-A binding protein binds to the early site and promotes the formation of a long UTR. So if we block this, we would expect that this site, the short site, is more likely chosen or more often chosen, and so we should see a reduction in the long UTR. So here's the control constructs, and now what we're doing is we're using a in situ hybridization probe that's specific for the long CAM kinase 2 3' UTR. So if you think about the overlay of the UTRs, there's a region in the long UTR that is specific for that long UTR, which we can detect with in situ hybridization probes. So here you can see the CAM kinase long UTR in 3 different, these are just representative images of cellular somata and dendrites, and here's an untransfected control. And then in the experiments where we've knocked down PABPN1, you can see that there's a dramatic reduction in both the soma and the dendrites of the long UTR. And this is also true if we use this point mutant version of PABPN1. So there's a reduction in the long UTR. And that's shown here, both for the soma and the dendrites, that there's less long UTR. Now, ideally, what we'd like to do is measure the short UTR, but there's no probe that you can design to detect the short UTR that won't also detect the long, because this is a sequence that they have in common. So to address the amount of the regular short UTR that we have, we use just simply in situ hybridization for the coding sequence. And what you can see here is that the knockdown of PABPN1 does not affect the coding sequence. So by implication, the short, there's a knockdown in the long, so these data suggest that there's actually an increase in the short 3' UTR. So it looks like the poly-A-BP binding protein is actually important in this system to favor the selection of the long UTR for CAM kinase 2. And we have similar data for some of the other family members that we predict should be PABPN1 targets. So this diversity that I'm describing by lengthening the UTR provides potential binding platforms for regulatory elements like RNA binding proteins that would then couple to the cytoskeletal machinery to drag the mRNA to the dendrites, or regulatory elements that would regulate the translation of the mRNA, for example, the addition of microRNA binding sites. And of course, the fact that you have this selection gives you all of the kind of potential that you have for alternative splicing. So you have the capacity during the processing of the transcript to make a choice about what features you want to endow that transcript with. So this raises the possibility that the 3' UTR choice could also be modulated by plasticity. And we're doing lots of experiments to look at this at the sequencing level, but I'll show you one experiment that we've done using the same knockdown strategy to look and see, for example, if 3' UTR choice might be important for plasticity. And in this particular experiment, the plasticity that we've chosen is to elicit homeostatic plasticity by blocking inhibitory transmission with bicuculline. And this is known to cause a downscaling of synaptic transmission, and this is typically measured by a decrease in the amplitude of the mini EPSC that you can see here. Now, we compared the effect of bicuculline in GFP alone transfected cells, or cells that were transfected with the short hairpin to knock down PABPN1. Here's the control. And what we found is that blocking the effect of PABPN1, reducing PABPN1, blocked the homeostatic plasticity in this case. Now, we have some sequencing data to suggest that the plasticity that's elicited by bicuculline results in a choice of shorter UTRs for some of the transcripts. So interim conclusions, I just want to show one other sort of technical thing. In the early data, I showed you that we've identified with deep sequencing and validated with in situ hybridization and nanostring thousands of mRNAs that are present in the processes. And now as we sort of dive in and look at the structure of these transcripts, we see that there's a lot of diversity and that the neuropil transcripts are different, not in the proteins that they code for, but in the way that they can be regulated. And this is accomplished by changing the structure of their 3' untranslated region, largely by elongating the UTR and providing more binding platforms for microRNAs and RNA binding proteins. And lastly, in the functional studies, I showed you that the way that this is accomplished during the processing of the mRNAs, at least for some of the transcripts, may be by a protein called poly-A binding protein, nuclear 1, which binds to the upstream poly-A sites, masks their choice, and results in the choice of the longer UTR in these experiments. So these studies all, getting back to the very beginning of my talk, I think address the potential of local translation. And then there's, of course, the issue of what is actually translated. And this is also something that my group is very interested in. And we've been working on techniques that will allow us to identify and visualize the proteins that are synthesized. And one of the techniques that we've used is using non-canonical amino acids to visualize proteins. This is a technology that we developed with Dave Terrell when my lab was still at Caltech. And so as discussed earlier today, these amino acids can serve as surrogates for cellular methionine, cross cell membranes, and get charged by the cell's own methionine tRNA synthetase onto methionine residues, and then get incorporated during protein synthesis. And the beauty of these non-canonical amino acids is that they have these functional groups, either azides or alkynes, which can be used as molecular handles using click chemistry. One can click onto an AHA-bearing protein, any kind of tag that you'd like. And so in experiments that we've done several years ago now, we showed that you could visualize newly synthesized proteins by using fluorescent tags. An example is shown here, where we showed that if you treated hippocampal neurons in culture with either AHA, an azide-bearing amino acid, or HPG, an alkyne-bearing amino acid, and then clicked with the appropriate tag, you could visualize, here shown in pink, all of the newly synthesized protein in this cell, or here shown in green. And all of this signal was dependent on protein synthesis. Now, this is nice, for example, in the experiments shown by Kim this morning, if one wants to ask, does a particular protein or agonist cause a change in protein synthesis in a specific cellular compartment, like the outgrowing retinal axons. But one of the things that always bugged us about this technique was that we couldn't really say what protein was synthesized. And we, of course, being interested in synapses, wanted to know not only is protein synthesis enhanced, but what particular proteins are being synthesized in which place. So the question is whether we could ever use this technique to actually say, a newly synthesized protein of interest has been synthesized, has been made. Let's not use synthesized too much. So we meaning Suzu Tom Dieck, has worked out a technique that couples this amino acid technology that we've worked on together with proximity ligation assay to do precisely this. And the way the technique works is it detects the coincidence of two antibodies. One antibody that we're going to use to recognize the protein as newly synthesized, and another antibody that's going to recognize our specific protein of interest. So the beginning of the experiment is just like we've said before. You metabolically label with a non-canonical amino acid. This amino acid gets incorporated into protein, and then using an antibody with an alkyne group, if this is an AHA molecule, and let's say it's a biotin antibody, you would treat with the antibody, and then you can recognize that biotin group with an anti-biotin antibody. And you can also recognize your specific protein of interest with an antibody. And then using the proximity ligation assay, you add secondary antibodies that have the PLA minus and plus strands on them, together with ligating oligos. And when these things are in close proximity, this whole thing can undergo rolling circle amplification and synthesize a strand that can be recognized by a fluorophore, so a labeled probe. So we've done many experiments, and I'll just show you a few, which allow us now to have a high degree of confidence that this actually works. So here, for example, is what we call FunCat PLA for CAM kinase 2. And this is, for what you see in green here, is all of the CAM kinase 2 that has been made in the last 2 hours. So here you can see green particles all throughout the dendrites, and here's a blow up here. You see it going through the dendrites, and you also see some in the proximity of the cell body. And if you do an experiment where you block protein synthesis and everything else is the same, this is the level of background signal that you can see, so that you can see there's a huge signal to noise. So why is this useful? Well, for example, many years ago when Mike Sutton was in my lab, He did experiments showing that local blockade of minis in neurons led to a homeostatic upscaling of synaptic transmission that was accompanied by more glutamate receptor on the surface. And that increase in the glutamate receptor on the surface was protein synthesis dependent. But we had no way of really saying whether it was newly synthesized glutamate receptor or some other molecule that was required for this process. And so now, for example, and these are Mike's data showing that under this perfusion spot where he locally perfused APV, he could see an increase in the surface GluR1 on, right under the perfusion spot that required protein synthesis. But now what we can do is directly, instead of just looking at total GluR1, we can say we're looking at newly synthesized GluR1. And here's an example of the experiment that we've done now looking at this GluR1 molecule under controlled conditions, under conditions where just action potentials are blocked, or under conditions where the NMDA component of minis are blocked. And here we can clearly see this increase that we hypothesized but couldn't directly show, and now we can directly show. We can directly see local synthesis. And actually, in this experiment, we can't say it's local synthesis. We can directly see new synthesis of the glutamate receptor. So in this last little part, I've just showed you the latest instantiation of the technology that we've been trying to develop that will allow all of us to look at newly synthesized proteins in situ. These are the people in the lab, and I'll point out Suzu Tamdik, who's the person who developed the technology that I just described. And the 3' UTR sequencing project and the original sequencing project was done by both Tristan Ville, who just defended his PhD thesis yesterday, and Georgi Tushev. Other members of that group are Gunay Akbalik and Irina Velatkovich, and I believe that's everybody on this slide. So thank you.